Welcome back to Human Biology with Dr. B. Today we're talking about the cell and the various organelles within. First things first, let's talk about the cell theory. There are three major parts to the cell theory. The first tenet is the cell is the smallest unit of life. As we've talked about, you cannot have life without having a cell. Even the smallest organisms are one cell big. Cells make up all living things. Every living thing has cells. Regardless of how big or how small it is, it's all made out of cells. And third, new cells arise only from pre-existing cells. Now, this may seem self-explanatory, but back in the 1800s, the prevailing thought of the day was that cells just spontaneously appeared. And so the cell theory and the video that I also have, the link is right here, but the video that I have on your homework also helps explain how we came to understand the cell theory as it currently is. So here's the cell. On the left, we have an actual micrograph of a cell. You can very clearly see the nucleus with the nucleic acid inside. You can see a number of the other organelles as well. And then over here on the right, we have what we refer to as, quote, a cartoon or a drawing depiction of the cell. And each of these organelles we will going through in detail through the next few minutes. First, let's talk about the plasma membrane. This is what delineates one cell from another. The plasma membrane is what we refer to as semi-permeable. It controls the movement of substances into and out of the cell. As we talked about with our macromolecule chapter, the plasma membrane is made of phospholipids. Now, phospholipids have a phosphate head and a lipid tail, which means that they have both hydrophobic, the inside, and hydrophilic, the outside portions. Now, the hydrophobic inner layer of the bilayer prevents hydrophilic molecules from moving through easily. The plasma membrane not only is made up of phospholipids, but also contains cholesterol, proteins, glycoproteins, and glycolipids. Now, the cholesterol helps maintain the fluidity of the membrane. The proteins, glycoproteins, and glycolipids can act as transporters, anchors, cell-to-cell recognition, as well as intramembrane enzymes. And here is a depiction of the fluid mosaic model. If you'll notice, each of these right here is a phospholipid, phosphate head, hydrophilic, lipid tail, hydrophobic. We have channels, we have various types of proteins. This is a channel protein. We have a glycoprotein over here. These act as cell-to-cell recognition or cell signaling molecules where cells can actually communicate with one another. You also see these cholesterol molecules. And these cholesterol molecules basically integrate into the membrane to help maintain fluidity. Because all of these phospholipids, they can move laterally, and sometimes recent research has suggested that they can flip side to side as well. But there are two faces to the cell, to the plasma membrane. We have the extracellular face of these plasma membrane, and we have the intracellular face of the plasma membrane. This becomes very important because on the intracellular face, these embedded proteins actually attach to what we'll talk about in a second, the cytoskeleton. And then the external side, the extracellular facing side of the plasma membrane, has all the cell-to-cell recognition and cell-to-cell communication, proteins and macromolecules. So then we move from the plasma membrane just slightly inward and we get to the cytoplasm. The cytoplasm contains the cytosol and organelles. So basically everything that makes up the cell on the inner side of the cell membrane is what we consider held in the cytoplasm. The cytosol is the jelly-like stuff within the cytoplasm that basically helps the cell maintain its three-dimensional shape. If we move a little bit further inward, we get to the cytoskeleton. The cytoskeleton is made up of a number of different proteinaceous filaments, if you will. We have microfilaments, intermediate filaments, and microtubules. And each of these three, just like our skeleton within the human body allows for us to maintain a shape, the cytoskeleton helps these cells maintain its various shape as well. Over here on the right, you can see this is a cell from your intestines. Now, the intestines, their whole job is to absorb nutrients. So they have developed this method of increasing surface area, and we call these microvilli. And these microvilli are finger-like projections on the external portion of the cell that allow for increased surface area so that as the food flows over the microvilli, or at this point it's called chyme, the fluid, we allow for maximal absorption and maximal surface area of exposure to the cell. Also, the cytoskeleton determines the location and synthesis of various proteins. Now, this becomes important not only, as we see here in these intestine cells, but also in various other cells throughout the body, because various functions within the cell need to have a number of different enzymes, and these enzymes definitely need to be located close together, so that way the function can be played out very quickly. So microfilaments, as we talked about, in the intestinal cells, we have these beautiful microvilli, and you can see the terminal web inside that creates this beautiful cytoskeleton. Now, these microfilaments are made of actin. They can also interact with myosin, as in the case of your skeletal muscles, in order to cause movement. These microfilaments, their main purpose is to anchor the cytoskeleton to the integral proteins in the plasma membrane. Integral proteins are proteins embedded in the plasma membrane, and when the cytoskeleton attaches to them, it basically holds them in place. So while the phospholipids might move within the bilayer, the integral proteins and the cytoskeleton do not change. And then we have intermediate filaments. I love how these are named. We have microfilaments, intermediate filaments, and then we will get to microtubules in a second. These are the intermediate size. So intermediate filaments, they strengthen the cell and help to maintain shape. They also can hold the organelles in position because, as we'll talk about, each organelle has a function, and each of these functions with regard to various types of cells contributes to the overall function of the cell. And when we position these organelles closer together or farther apart, it helps contribute to the overall function of that cell. Also, it stabilizes the position of a cell within a tissue. So intermediate filaments help solidify or basically hold more than one cell together so that each cell performs its function, and then on a tissue level, the tissue, all of the cells grouped together, can perform their functions as well. And then we get to microtubules. They're made of tubulin, and microtubules play a large role in cell division. They extend outward from the centrosome. The spindle will attach to the chromosomes during cell division and will help the chromosomes move to opposite ends of the cell. They also help maintain the shape of the cell, and they form a majority of the cytoskeleton, and they can help move organelles and vesicles. So here's the centrosome, and you can see extending from there the microtubules. The centrosome I like thinking of, and we'll talk more about this as we get into cell division. The centrosome basically is this larger organelle that acts as a fishing rod. So these, and this is the reel, we call it the centriole in the middle, and then from it extend microtubules. And the centriole acts on the microtubules kind of like a fishing rod, while the microtubules will attach the chromosomes, and they will push and pull to the opposite sides of the cell in order to create two new cells. And then we get to the nucleus. The nucleus, most cells contain only the one, but there are a number of multinucleate cells in your body. Your skeletal muscles are one of such examples. Within the nucleus, they have nuclear pores that are necessary for, it says, chemical communication, also communication with nucleic acids. And outside, there's a double membrane that we call the nuclear membrane, or the nuclear envelope. And then inside, it's separate from the cytoplasm because the nucleus is the largest organelle within the cell, or it's the easiest seen, if you will. It's this big globular round ball in the center. And within it, it has the nucleoplasm. Separate from the cytoplasm, it's only contained within the nucleus. And it keeps its shape, the nucleus keeps its shape due to the nuclear matrix. Within the nucleus's main job is to hold and contain all of the chromosomes that make that cell part of the larger organism, or that cell an organism. Now here you can see on the left individual chromosomes, and then on the right you can see how large the nucleus is in comparison to the rest of the cell. And you can see the chromatin, which is basically relaxed chromosomes with associated proteins, as it takes up the majority of the inner part of the nucleus. Then we move externally to the rough endoplasmic reticulum. The proteins are made within the rough endoplasmic reticulum. These are primarily made for excretion. Some can be made for within the cell, but primarily these will be excreted outside the cell. We call this the rough endoplasmic reticulum because, as you can see down here, the endoplasmic reticulum is part of the endomembrane system. The endomembrane system consists of the nuclear envelope, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi, and any associated transport vesicles. We'll talk about those in a moment. But here you can see on the external portion of the rough endoplasmic reticulum, we have ribosomes. Ribosomes are not organelles. They are RNA and some protein that allow for the making of proteins, other proteins. And they embed themselves on the external portion of the rough endoplasmic reticulum, and they make their proteins inside, and then these proteins will be moved into a transport vesicle. And here you can see the endomembrane system much better than what I just described. So here is the nucleus. You can see the nuclear envelope, and the nuclear envelope is continuous. Another name for continuous I like using is called contiguous with the rough endoplasmic reticulum. And then you can see out here the smooth endoplasmic reticulum. Now, these are genius names because the rough endoplasmic reticulum, when you look at it under a microscope, looks rough because of the presence of ribosomes. And the smooth endoplasmic reticulum looks, wait for it, smooth because it does not have the sandpaper-ish quality of ribosomes. Now, the smooth endoplasmic reticulum also smooth because it makes phospholipids, cholesterol, steroids, etc. Also, it detoxifies drugs. So a great question would be, what cells in your body would have a lot of smooth endoplasmic reticulum? Think about what organs detoxify your body. It also, in other specific types of cells, specifically any type of muscle cell, it stores calcium. Yeah. And then we get to the Golgi apparatus, also part of the endomembrane system. This is where proteins are modified and packaged for secretion. I like thinking of the Golgi apparatus like the UPS store. So what do you do when you go to the UPS store? You bring your thing that you want packaged, and you go up to the counter at the UPS store and you say, hey, I need to send this to X, Y, and Z. And they say, okay. And they put some packing peanuts in it. They wrap your thing in bubble wrap, and then they put it in a box, and then they tape the box closed, and then they put a label on it. And the label tells you where it's going to go. The same basic things happen in the Golgi. This is where proteins are modified, so like the bubble wrap and putting packing peanuts around, and then they're put in what we call a vesicle, which is more or less like the box in which they put it. And on the box, also on the protein, there are what we have, they put on there what we call a label. And the label basically tells the cell and all the cell machinery if this is going to the cytoplasm, if this is going to the cell membrane, what portion of the cell membrane, where in the cell, etc., etc. It's kind of genius. The Golgi also produces lysosomes. Lysosomes contain lysozyme, which is an enzyme that lyses larger proteins, as well as it contain hydrolytic enzymes and hydrolytic chemicals that will basically break things down. And here you can see again, we have the endomembrane system, we have the nuclear envelope, we have the rough endoplasmic reticulum, we're missing the smooth endoplasmic reticulum somewhere over here, and then from the rough endoplasmic reticulum, these transport vesicles they bud off of the rough endoplasmic reticulum, they then go to the Golgi, which basically looks like a stack of pancakes, also the UPS center, where these proteins are packaged, modified, and then given a label, a shipping label, and in most cases, they will come out here and become a secretory vesicle, where they will, the external portion of the membrane that budded out from the Golgi will then become part of the cell membrane, and the protein products are then released from the cell. Lysosomes, talked about this. 40 different digestive enzymes. Lysosomes are very important, as we saw in the hidden life or the hidden universe life of the cell. They fuse with damaged organelles to break them down. And this also plays a large role in autolysis or autolysis, which is internal digestion of the cell, which occurs when the cell becomes diseased with a virus or just basically gets old and broken down. This can also happen with organelles. You can see this here. So here we have a damaged organelle. It will fuse with a lysosome. The lysosome enzymes will break down the organelle and then release the products or release the portions that have been broken down back into the cell for recycling. And then over here we can have, this is what we call phagocytosis. A bacterium is ingested by the cell. It binds with a lysosome. This becomes a phagolysosome. It breaks up part of the invading bacterium or the virus and releases it externally. The mitochondria. Now, the mitochondria, as you may have heard, is the powerhouse of the cell, also known as the respiratory center of the cell. The mitochondria has its own DNA. We call it mtDNA or mitochondrial DNA, and it is inherited from your mother. It also has its own ribosome so that it can make its specific enzymes and proteins necessary for cellular respiration. The mitochondria, due to its high acidic content within the inner portion of the cell and this high hydrogen ion concentration, high acidic concentration, is what powers ATP synthesis. So there's a double membrane that keeps all of these hydrogen ions on the inner side, on the what we call the matrix of the mitochondria. It's actually in the intermembrane space. And here you can see the mitochondria. This is an actual micrograph. Here is a cartoon. You can see the mitochondrial matrix on the inside. You can see the cristae. You can see the intermembrane space. The intermembrane space is where the hydrogen ions are pumped in order to make ATP as they flow back down the gradient. Pretty awesome. On cilia and flagella. Cilia, you may know from your bronchi or from your trachea. Whenever you get a cold, and your nose starts running, you get postnasal drainage, and this is literally what happens. It drips down the back of your throat. Well, your cilia catch it and are like, not today, not today. And your cilia push all of that mucus back up your trachea and into your throat, which is what causes that gagging reflex as well as coughing. And now I feel sick just talking about it. The primary cilium it acts as a cell center to environmental stimuli such as postnasal drainage, sick and gross. And then we have motile cilia, which moves fluid and secretions. Now there's a number of places in your body that you have cilia. Number one, your trachea and your bronchi. And in women, fallopian tubes, also known as the uterine tubes, they move the ovum to the uterus. And cilia look like the microvilli that we see in our gut, but they are not the same thing. And then we know flagella from sperm. There they are. These provide the movement of little spermies. And that brings us to the different parts of the cell. Now you should be able to list all of the major organelles in the cell, describe how the structure is related to the function of each organelle, and how each organelle can help contribute to the overall function of the cell. It's been my pleasure. It's been another episode of Human Biology with Dr. B. See you next time.